Good morning, children. Last class I have discussed with you about the incomplete dominance. It is a type of intragenic interaction, and now I am going to discuss with another example for intragenic interaction, that is called codominance. What is mean by codominance? What is mean by codominance? One, alleles of gene expressing different alleles of a gene expressing their phenotype together, and it is called codominance. In that, neither of the alleles will dominate over each other, right? And this is called codominance. For this, AB blood group is an example of codominance. Okay, in AB blood group, the gene of AB blood group consists of two alleles. One is A, another one is B. In that, neither A or B is dominant over each other, but together they express their phenotypic character in the F1 generation, and this is called what? AB blood group, and it's called, this type of dominant is called codominance. Okay, this happened in the animals. Here in plants, we will see another example. This pattern occurs due to simultaneous expression of both alleles in the heterozygote. Okay, the phenomenon in which two alleles are both expressed in the heterozygous individual is known as codominance. Heterozygous individual means what? The phenotype, the alleles for phenotypic characters will not be same, but it is differ. But when though it is differ, the two characters are expressing together in the young one, and that is called what? Heterozygous. Example in flowers: red and white flowers of camellia, inheritance of sickle cell hemoglobin, ABO blood group system in human beings. These are example for codominance. In human beings, IA and IB alleles of I gene or codominant, which follows Mendel's law of segregation. The codominance was demonstrated in plants with the help of electrophoresis or chromatography for protein or flavonoid substances. Example: Gossypium hirsutum and Gossypium stortatum. Their F1 hybrid was tested for C proteins by electrophoresis. Both the parents have different banding patterns for their seed protein. In hybrids, additive banding pattern was noticed. Their hybrid shows the presence of both the types of proteins similar to their parents. Okay, so the heterozygote genotype gives rise to a phenotype distinctly different from either of the homozygous genotype. The F1 heterozygous produces a F2 progeny in a phenotypic and genotypic ratio of 1:2:1. So this what happened. Next, lethal gene. An allele which has the potential to cause the death of an organism is called a lethal gene. Okay, and what is mean by lethal? Lethal means death. When allele causes death of an organism, and this is called what? Lethal gene. What is lethal gene? 2 mark question. An allele which has the potential to cause the death of an organism, and it is called what? Lethal allele. In 1907, E. Bohr reported a lethal gene in snapdragon. It is an example for recessive lethality. In snapdragon, there are three kinds of plants. In snapdragon, there are three kinds of plant. One is green plants with chlorophyll, and its allele is capital C, capital C. Another one is yellowish green plants with carotenoids, or referred to as pale green, golden or aurea plants, for its allele capital C and small c. And another one type, white color plants without any chlorophyll, and its allele is small c, small c. So in snapdragon plants, It is an example for lethal plant. It has three kinds of plant. One is green, another one is yellow, another one is white. The green plant possesses chlorophyll, white plant without chlorophyll, yellowish plant which possesses carotenoids, and it's referred as pale green. And the gene alleles also differ in each cases. For green, alleles are capital C, capital C. For yellowish green, alleles are capital C, small c. White plant, the alleles are small c, small c. The genotype of the homozygous green plant is capital C, capital C. The genotype of the homozygous white plant is small c, small c. The Aurea plants have the genotype capital C, small c because they are heterozygous of green and white plant. When two such Aurea plants are crossed, the F1 progeny has identical phenotypic and genotypic ratio 1:2:1 with one green, two Aurea and white. Since the white plants lack chlorophyll pigment, they will not survive. So the F2 ratio is modified into 1:2. In this case, the homozygous recessive genotype C, small c is lethal. Okay. Actually, it is like a monohybrid cross only, children. In Snapdragon plant, as I said, it has three types of plants: green, yellowish green, and white plant. Both green and white have a homozygous allele. Green has capital C, capital C. Both alleles are same, that is homozygous. And the same way in white plant, the alleles are small c, hence it is also homozygous nature. But in F1 plant, that is Aurea, it consists of heterozygous allele, that is one capital C, one small c. So when this Aurea are self-crossed, that is when two Aurea plants crossed with each other, as usual monohybrid, it produce three kinds of genotype. One is capital C, another one is capital C, small c, another one is small c in the ratio of 1:2:1, right? Okay. Among this, the white plant, since it does not contain any chlorophyll in it, it cannot do photosynthesis, and the plant with white color will die. So this is due to the lethal gene small c, which is recessive in trait. Okay. So this recessive trait responsible for the death of organism, hence it is called what? Recessive lethal gene, right? Okay. Since the lethal genes have died, lethal plants are died, this white plants are died, and the ratio of the F2 generation is 1:2. Okay. So that's what given here, you can understand with that, children. The term lethal is applied to those changes in the genome of an organism which produces effects severe enough to cause death. Lethality is a condition in which the death of certain genotype occurs prematurely. The fully dominant or fully recessive lethal allele kills the carrier individual only in its homozygous condition. So the F2 genotypic ratio will be 2:1 or 1:2 respectively. Okay. The term lethal is applied to those changes in the genome of organism which produce effects severe enough to cause death. So the lethal gene which cause changes in the genome, then the genome of organism produce severe effect which cause the death. Lethality is a condition in which death of certain genotype occurs prematurely. Then the fully dominant or fully recessive lethal allele kills the carrier individual only in its homozygous condition. So lethality is happen in the homozygous condition only. That may be either in the fully dominant condition or either in the fully recessive condition. In that condition only the lethal genes cause death. So the F2 genotypic ratio will be 2:1 or 1:2 respectively. Next, pleiotropy. a single gene affects multiple traits is called a 2-mark question, children. Pleiotropy. What does mean by pleiotropy? As of Mendelian genetics, one gene control one character, right? But here many single gene affect multiple traits. Single gene affect multiple traits. That is called what? Pleiotropy. In pleiotropy, the single gene affect multiple traits and alter the phenotype of the organism. The pleiotrophic gene influences a number of characters simultaneously, and such genes are called pleiotrophic gene. Okay, the in pleiotrophic characters, single gene affect many characters simultaneously, and such genes are called pleiotrophic gene. Were crossed with a variety of peas having white flowers, light colored seeds, and no spot on the axil of the leaves. The three traits for flower color, seed color, and a leaf axil spot all were inherited together as a single unit. Another example is single sickle cell anemia. Okay. See, were crossed with each other, pleiotrophic genes were crossed with a variety of peas having white flower, light colored seeds, and no spot on the axil of the leaves, and three traits for flower color, seed color, and a leaf axil spot all were inherited together as a single unit. So, in a pea plant, all these characters are inherited together as a single unit. Another example for pleiotrophic is sickle cell anemia. So here in this, children, you will get as a 2-mark or 3-mark question. What is pleiotropy or and what is pleiotrophic gene? Give example. Okay, it is a 2-mark question or 3-mark question you will get. Okay, bracket it. It is important also. And intergenic gene interaction. So far we have discussed about intragenic interaction. Under this we have discussed 3 subtopics. One is incomplete dominance, another one is codominance, another one is lethal gene. And now we are going to discuss about intergenic interaction. Okay, intergenic gene interaction. Intra means what? Alleles of the same locus. Interaction among the alleles of the same locus. That is called intralocus interaction. Here interlocus interaction, that is alleles, the interaction between alleles of two different loci is called what? Intergenic gene interaction. So let's see, interlocus interaction takes place between the alleles at different loci. That means one allele may be present in one place of the chromosome, another pair of allele present in the another locus. Locus means what? Where the genes are located in the chromosome is called loci. Each gene has its own specific place in the chromosome. It is constant to each gene in a chromosome, and that particular place is called loci. That means in the chromosome genes are present in a particular loci, and that loci is called what? Particular place, and this place is called loci, right? Okay. And here between alleles of different genes, if in different genes, it is called what? Intergenic interaction. Intergenic interaction includes dominant epistasis. Okay, what does mean by dominant epistasis? Dominant epistasis is nothing but when one pair of allele present in some loci of chromosome will suppress or mask the expression of phenotype of alleles present in another locus. It is called what? Dominant epistasis. அதாவது ஒரு குரோமோசோமோட ஒரு பர்டிகுலர் பிளேஸில் இருக்கக்கூடிய ஒரு பேர் ஆஃப் அல்லீல் இன்னொரு லோக்கையில் இருக்கக்கூடிய இன்னொரு பேர் ஆஃப் அல்லீலோட ஃபீனோடைப்பிக் எக்ஸ்ப்ரெஷனை சப்ரஸ் பண்ணுறது அல்லது மாஸ்க் பண்ணுறது. And this is called what? Dominant epistasis. This is also a 5-mark question and important too. Dominant epistasis, it is a gene interaction in which two alleles of gene at one locus interfere and suppress or mask the phenotypic expression of a different pair of alleles of another gene at another locus, right? This is called what? Dominant epistasis. 2 mark question. Sometimes you will get question like explain intergenic interaction with example அப்படின்னு ஒரு கொஸ்டின் கேட்டுருவாங்க. ஸோ இன் தட் கேஸ் யூ ஹாவ் டு எக்ஸ்பிளைன் வாட் இஸ் intergenic gene interaction, and example for intergenic interaction is dominant epistasis, and you explain that. Okay, so next one, the gene that suppresses or masks the phenotypic expression of a gene at another locus is known as what? Epistatic. Okay, the gene which one is suppress or express in the phenotypic of other gene is called epistatic, and the gene whose expression is interfered by non-allelic gene and prevents from exhibiting its character is known as hypostatic, right? So simply if we speak, the gene which express the character of other gene is called epistatic. The gene which is suppressed by epistasis is called hypostatic. That's all. When both the genes are present together, the phenotype is determined by the epistatic gene and not by the hypostatic gene. Okay, in a condition when two genes are present together, that is both epistatic allele and hypostatic allele present in the same locus, in that case what happen? The epistatic character will be expressed and hypostatic character will be suppressed. That's all. In summer squash, the fruit color locus has a dominant allele capital W for white color and recessive allele small w for colored fruit. Okay, so fruit color is white and recessive for capital W for white color and recessive allele small w for colored food. So if if the gene possesses capital W, then the fruit color will be white, and if the gene possesses small w, and the color of the fruit will be different, okay, maybe colored. W allele, capital W allele is dominant that masks the expression of any color. In another locus, hypostatic allele capital G is for yellow fruit, and it is recessive allele is small g for green fruit. In the first locus, the white is dominant to color, whereas in the second locus, yellow is dominant to green. When the white fruit with the genotype capital W small g crossed with yellow fruit with genotype small w capital G, and the F1 plants have white fruit and are heterozygous capital W capital G small g. When F1 heterozygous plants are crossed, they give rise to F2 with the phenotypic ratio of 12 white and 3 yellow and green. Okay, right? So here what happened? White, we have taken two types of plant. One in first cases capital W white and small w for colored fruit. So here capital W dominant over small w, and when both alleles are present, capital W express white color and the small w's colored fruit will be suppressed. And in another locus there is an capital G, it's express the yellow in color, and small g which is for green in color. In this case capital G is, that is yellow color dominant over green. When we cross these two plants, that is capital W small g as one parent, and with white color fruit, and another one is small w capital G, it express yellow color. இதுலயே நம்மளுக்கு தெரியுதுமா, கேபிட்டல் G இருக்குது, ஸ்மால் w இருக்குது, பட் கேபிட்டல் ஜிக்கான எல்லோ கலர் தான் எக்ஸ்ப்ரெஸ் ஆகிருக்குது, ஸ்மால் w கான கலர் எக்ஸ்ப்ரெஸ் ஆகலை. அப்போது இங்கே என்ன நம்ம சொல்லலாம்னா கேபிட்டல் ஜி இருக்கிற இடத்துல ஸ்மால் டபிள்யூ ரெண்டுமே இருந்ததுன்னா கேபிட்டல் ஜியோட கேரக்டர் தான் எக்ஸ்ப்ரெஸ் ஆகும் அப்படிங்கிறது நம்ம தெரிஞ்சுக்கலாம். ஸோ ஓகே, F1 ஜென்ரேஷன் எல்லாமே ஒயிட், all are white in color. Capital W, small w, capital G, small g. So heterozygous gives rise to white color fruit only. None of the other color will be expressed in the F1 generation. So from that we conclude if whether it will be small g or capital G or small w, whatever the alleles are present along with the capital W which encode for white color, only the white color will be expressed, that is capital W dominant over the all other colors, right? Okay, so then what, when F1 are self crossed, what will be the result? There is a 12 white, 3 yellow and 1 green. That means capital W dominant epistosis over other character, capital W, that is white color. The allele capital W dominant or epistotic over the other colors. Okay, that's what given here, checkerboard children. See, 12 yellow white color and 3 yellow color, 1 green color will be the ratio. Since capital W is epistotic to the alleles capital G and small g, the white which is dominant masks the effect of yellow or green. Then homozygous recessive small w genotype only can give the colored fruit, that is 4 out of 16. Double recessive capital W small g will give green fruit, and in the ratio of 1:16. So the plants having only capital G in its genotype, that is small w capital G small g, or small w capital G will give the yellow fruits in the ratio of 3:16. That's what given in the checkerboard also, children. Please go through it. And intragenic or allelic interaction and its ratio, incomplete dominance. Example for incomplete dominance: flower color, already we have discussed Mirabilis jalapa, flower color in Snapdragon, both also 1:2:1, in both 1:2:1, and in codominance ABO blood group also 1:2:1. And in intergenic or non-allelic interaction, epistotic interaction, dominant epistosis. Example for dominant epistosis: fruit color in summer squash, its F2 ratio is 12:3:1. And recessive epistosis, flower color of Anderinum, that is Snapdragon species, 1 is 9:3:4. Duplicate gene with cumulative effect. These all we did not have in our syllabus. Simply they have given the type and its ratio. Example alone they are giving. Duplicate gene with cumulative effect. Example, fruit shape in summer squash plant, and its ratio 9:6:1. Complementary gene. Example for complementary gene, flower color in sweet peas, and its F2 ratio is 9:7. Supplementary genes, grain color in maize, and its F2 ratio 9:3:4. Inhibitor gene, leaf color in rice plants, and its ratio 13:3. Duplicate gene, seed capsule shape in shepherd's purse, Bursa pastoris, and its F2 ratio is 15:1. Okay, children. With this we completed the genic interaction. These two, or whether it may be intragenic interaction or intergenic interaction, will be considered as extension of Mendelian genetics. And next one is polygenic inheritance in wheat. This I will discuss with you in my next class. And your homework will be dominant epistosis and codominance. These are the two questions I will give as a homework, children. Complete it. Children, please, you will get exams very soon. Monthly test, Jan month test you will get very soon, and prepare yourself, okay? And I will get you in my next class. Bye, children.